So today I want to thank Michael Snyder, the chair of our event today. Did his PhD at Caltech, was a professor at Yale, and has been at Stanford for 15 years, is it? Been following his work for a big chunk of that time, actually, to study the systems biology of aging and in the level of detail that we need to be looking at it, not just these single-dimensional biomarkers. Allison?
Well, I think the interesting thing about you is that you're both a subject and a researcher in some of these studies. And when we had a brief time to chat at the sponsored drinks, you pointed out a few other studies that you were about to do or are in the process of doing, where I've always wanted to know the answer to. So I'm really, really excited to hear. Hopefully, you can spill the beans a little bit on what's ahead as well. You've also, really helped really co-found and advise many different companies, consortia, and so forth. And thank you so much for doing this event with us. You really you're living what you preach. So thanks for that. With that, without further ado, please come up on stage.
It is a pleasure to be here. I'm happy to chair this session, but I didn't nominate myself to be a keynote. But anyway, thank you for that. Thinking of reflecting a little bit about the aging field in general, at least when I first became aware of it, it seemed like it did originate in academia, and it was very, very focused on mechanistic studies. How do organisms age at model organisms in humans? What are the mechanistic processes? Then when thinking about translation, it really seems like that emerged more recently. I would argue perhaps in the last decade, maybe before, and that's mostly centered from the startup area, I would say. And then where I come in really is from the wellness side of things, how to extend health span and turn, I guess, lifespan. And to a large extent, these started out, I would argue, largely separately, although I think over the last several years, it feels like they are merging quite a bit. And I think these Venn diagrams will probably become completely entwined if things continue the way they're going, which I actually think is a really good thing to build an ecosystem like Foresight and other groups here are trying to do. I think will be very, very powerful for really getting maximal impact. So as I say, we come from the wellness field, and I like to say that healthcare system is broken, for those of you who've seen me talk before. And it really is sick care, not healthcare, and the whole financial incentives are set up in alignment with that. That is, people usually go to a physician when they're sick, and that's how the physicians get paid. But we do, I think all people in this room would agree, we really do need to switch things to more preventative medicine or precision health, if you want to call it that. But even the way we practice it today is pretty antiquated, and I would argue all these steps can be changed. Let's see, I don't know if there's a pointer. There's one. Let's see if this works. Yeah, so can you see that? You want me to use the arrow instead? Problem is I always wind up switching slides when I try that, so let's see. So far I'm being unsuccessful. Yes, there we go. I rest my case. All right, I'm going to use this, even if you can't see it very well. Sorry for you guys over there. But basically, the idea, when you think about how you practice healthcare now, well, what you'll do is you'll typically get in a car in the U.S., the travel physician's office, always at an inconvenient time. When you arrive there, the office looks pretty much the same, except for a few new gizmos. When you're there, they'll stick a needle in you and draw lots of blood. And from all that blood and the time you're there, they actually don't make that many measurements when you get right down to it. And then from those measurements, they'll treat you based on population averages. So we're actually trying to change all of these steps, as you'll see. And with regards to this last one, I'd like to show this, and some of you I'm sure have seen this before, but we've been told since we're little, our temperature when you put a thermometer in your mouth is 98.6. But if you look at the data that are out there, there's a number of studies, and they all tend to come out more like this, 97.5. It actually drops as you age, believe it or not. That's true for me. And so, first of all, it's not what we've been taught, but the more important point, of course, is there's a spread. So this is 25th quartile, 94.6, 75th quartile, 99.1. And so what that means in today's world is that if this is your normal healthy baseline at 94.6, you go to a physician's office, they measure you at 98.6, they'll tell you you're normal, you're healthy, everything's fine, go home. But the reality is if you're up 4 degrees over your baseline, I guarantee you're not healthy. Something is off. And so that's the big theme of our work, collect a lot of data around your healthy baseline so you can detect disease shifts. Now, your health, as I'm sure you appreciate, it's influenced by many things, and we are actually trying to measure many of these things. Your genome counts for about 16% of your lifespan, as many of you may know, although very big error bars in that. But all these other things we know intuitively impact your health, your exercise, food, mental health, environmental exposures. And we're in a world where we can measure all of these things, some pretty well now. Activity, for example. Food is still pretty clunky, but it's doable. Mental health, we tend to measure indirectly, and that probably has multiple dimensions to it. And then we actually are spending a lot of time on these environmental exposures, which have been brought to the forelight with COVID. But even if you don't measure these directly, of course, you can measure them indirectly by actually, and perhaps even more precisely, by trying to take deep measurements on people. And that's thanks to this revolution in technologies. We can now measure people in exquisite detail. So we can sequence genomes. The reagent costs about $100, although interpretation's more than that. But you can commercially get from Myome or Nebula. I have no conflict. I should say I'm conflicted on everything I'm going to tell you about today, except for this one. I'm not conflicted on, well, I have a sequencing company. It's not Myome or Nebula, but they'll sequence your genome for about $500 and give you some limited interpretation. But we would argue mass spectrometry a little bit quieter, but you can measure thousands of analytes out of blood and urine. And of course, the wearable revolution and other technologies have also lets us bring in all kinds of data as well. And so as many of you know, we set up this concept of doing personal omics profiling on people many years ago, 14 on me, and then a little over 11 years on a smallish group of people, 109. It's sort of expanded to 200. A few have dropped off, but we add more to keep the number roughly constant. And the idea is we'll sequence people's genomes once, and then we'll make many, many other measurements, mostly out of blood, peripheral blood monocyte cells for epigenome, which is methylation, transcriptome, proteome. And then cytokines and proteins and metabolites and lipids will measure out of plasma. We've followed 4 different microbiomes now: gut, nasal, tongue, and skin. I won't talk so much about that today, although I had a very nice study out on this. And some of these things will measure out of urine as well, metabolites and lipids, and the microbiome from those multiple sites. And then wearables, we started about 10 years ago when they came out as fitness trackers. And then this is on top of deep clinical tests, questionnaires, and some advanced tests like stress echocardiograms, and a lot of glucose control measurements. We're going to spend some time on that. I'll show you some new data there. And one reason for that, again, another conflict here, I'm type 2 diabetic, I'm a weird one, but figured out through deep data profiling, actually. So one aspect is take very, very deep data on people, and the other aspect is do it longitudinally. So we'll collect samples every 3 months while people are healthy. And then if an adverse event like a viral infection comes along, we usually take 5 to 7 more samplings across about a 5-week period. And we'll measure other times as well. And so you might say, why are we doing all this? We're trying to understand what a healthy profile looks like, not a sick profile. How does it change over time? Very relevant for aging. What's the difference between you and the person sitting next to you? I won't talk a whole lot about this, but the reality is you're more different from each other than a healthy you from a sick you is. And that's why it's really important to get your baseline profile. Yeah, and then we also want to understand, could these advanced technologies be used for better managing health? And I can tell you when we started this, most physicians did not like sequencing genomes of healthy people. They thought that was a terrible thing to do. I even had one yell at me for doing this because they said, you're going to turn everybody into hypochondriacs, going to cost millions of dollars, is a really bad idea. I think they've largely warmed up to that now, although I can tell you the wearables are not very well received by most physicians as well. We'll warm them up to that over time, I'm sure. All right, so anyway, just on the first, with regards to this point about can these advanced technologies be used for health monitoring, what we discovered from the first 3.5 years, there's more now, I should update this, but we have about 49 what we call major health discoveries, a number of minor ones as well, and it spanned all these different areas, so like hem-oncology, cardiovascular disease, and so on. And some of these were a big deal. We caught some with early lymphoma, 2 people with pre-cancers. I should say these are all found pre-symptomatically, so before folks had symptoms. And then, for cardiovascular, we had 2 people with serious issues here. One was picked up by genome sequencing, another through wearables. And that's another point. No one technology found these things. Sometimes it's genome sequencing. Sometimes it was the omics, the biochemical side of the omics. Sometimes it was imaging. Usually it's multiple data types, like the lymphoma was picked up by imaging plus some biomarkers that were off, some cytokines. And so that suggests that things were off. And so again, the idea is that you could get, by having a much deeper data dive, get a bigger picture of people's health. And the analogy I like to use is that if your health is a 1,000-piece jigsaw puzzle, we're trying to get more like 700 pieces, where we would argue getting about 5 or 6 today. You get a very, very incomplete picture with today's technologies, as it's currently practiced. So genome sequencing was powerful. Just from the first 70 people, 12 had pathogenic mutations that were worth looking at. These are single gene mutations, Mendelian, if you will, that lead to high propensity of disease. And many of you, I'm sure, know BRCA mutations. Women with BRCA mutations, very high frequency, will get breast or ovarian cancer. And so knowing this is a good thing. These others, most of them are lower frequency penetrance, meaning less chance of getting this. But 3 of them actually turned out to be a big deal. One individual with the mutation here actually puts him at high risk for heart problems, and his father died in his 60s of a heart attack. And sure enough, he did a follow-up based on the genome sequence, and actually has a heart defect. And so we think that mutation is the culprit. Another individual had a mutation here. There were 2 of them actually. Puts them at certain risk for high cancers. They did a whole body MRI follow-up, and sure enough, they actually had early thyroid cancer. That was removed, and they were able to keep most of their thyroid. So that was useful to find. And the 9 individuals are thought to be type 2 diabetic coming in. One is actually not a typical type 2. They're what's called a MODY mutation. They had an HNF1 alpha, which is a different kind of diabetes. And we'll talk more about diabetes in a minute. But basically, they were on the suboptimal medication for years because they were treated like a typical type 2 diabetic. So they shifted after this. So it's a minority of people, but if you're one of these 12 out of the 70, it was kind of a big deal. So most genetic diseases, you probably know, they're not single gene mutations. They're complex disease. And the way this is thought then to occur is that you have common variants. We have 4 million so-called common variants that differ between us and the reference genome. And the idea is that if you inherit a lot of the common variants that are bad, you're at high risk, and if you get the good ones, you're at low risk. And so people have built these polygenic risk scores to try to actually decipher this. Here's the analogy here, that a single gene variant like a BRCA, you get clobbered by a sledgehammer, whereas these common variants, they're actually thought to be of low effect. And so if you get, again, lots of bad, people typically integrate now over thousands or millions. And the idea is they have small effects, but they add up and ultimately can be deleterious. And so people have built these polygenic risk scores. Have you heard of polygenic risk scores? Yeah, this crowd generally knows about it. So what they do is they sum over thousands or millions of variants to make predictions about your genetic risks. And it turns out it works for about the top 2 to 4%. If you look at what's called the odds scores, that sort of, it just works for that group. It really doesn't work for most people. And does anyone know why? Or what are your theories why a polygenic risk score is not so good? Well, it turns out, I'll give you my theory with 2 reasons. One is that genetics isn't linear. They do linear addition across these thousands of variants. And anyone who's done genetics knows that 1 + 1 is rarely 2. If you have 2 mutations, same biochemical pathway, 1 + 1 is 1. You don't get additive effects. If you have 2 mutations, different pathways, 1 + 1 can be 15 in terms of effect size. So the point is that genetics isn't linear. It also ignores all the rare variants that are probably contributing to this as well. So I won't have time to get into this, but you can look up the papers. We've come up with new ways with machine learning, which turns out to be very, very powerful because it's nonlinear. It lets you grab groups of genes in a nonlinear fashion, for those of you who know what I'm talking about. And it turns out it works. You can now get at the genetic basis of diseases much better with this approach. And we use 2 different approaches. I won't have time to get into this, but basically for this one, abdominal aortic aneurysm, people usually discover they have it when their aorta bursts and they die, which is a terrible way to know you're at risk, right? You want to know ahead of that. And so there were 3 questionable genes, and using a machine learning approach and pathogenic variants, we came up with 60 genes, which we're pretty confident most are right, and went on to validate them. And then for these other cases, these are all known to be heritable, by the way. ALS is 61% heritability. AA is about 70%. Here there was a big, they're called genome-wide association studies, where 7 genes have found. With this new machine learning method, we came out with 700. We could explain from, instead of 6% of the heritability from this, we got 36%. And then severe COVID, you may know a lot of young people get severe COVID and get hospitalized and die. And that's thought to have a genetic component associated with it. So there was a giant GWAS study that had been published, and we took that same information, did this new machine learning method, and came up with, and single cell data, and came up, went from 47 genes to over 1,000. Here we can explain for the most severe form of severe COVID, 77% of the heritability, which is like unheard of. So you can capture a lot more of the genetic information with this kind of new approaches. And yeah, sorry not to go in this more detail, but happy to talk with you later about it if you like. So I think this will be promising. In this first case, we even could come up with a predictive score that's kind of decent, that from the genome alone, we can make some prediction about your risk. And with genome plus EHR data, you can do even better. This is early days. I know we can do better than this. We're not there yet for ALS or for severe COVID, although we can predict prognostics for both of those, like how severe the disease will be. So, but I know we can get there with, I'm confident we can get there with prediction of who will get it. We just need more information. So the other thing we learn about all this is that as we have profiled people over time, of course, we've noticed that they are changing through these omics profiles. And one of the things we first discovered is that, in fact, we're interpreting this as aging, and that everyone is aging differently. So we had about 600 markers that changed significantly over time. And this person, their top biochemical pathway that changed was this cardiac hypertrophic signaling pathway. So in fact, they're a cardioager, if you will. This person over here happens to be me. Their immune system, metabolic, and other pathways change with time as well. Pretty typical ager. And so in the end, we could classify people into what we call ageotypes. And at the time, we were only empowered to find 4. We know there are many more: kidney, liver, metabolic, and immune. And so for each row is an ageotype. Each column is a person. And so the 4 people over here, they're aging in all 4 categories. The box over here, they're aging in 3 of the 4, not much of a kidney ager. This person's kidney, not much of the others, kidney and a metabolic, and so on. So you can see how people are aging. And then it turns out there are actually clinical markers associated with these ageotypes, so you can act on those. And some people, I won't show you this, have, especially in the metabolic category, they changed their lifestyle, either took metformin or exercise, and they shifted their ageotype in a favorable fashion. Now, I'm not saying they got younger, but they had better biochemical markers, which was nice to see. So we thought about this more in more detail. And if you think about the way most of these markers are gained is using linear regression models. So like looking for how things change. And when people do these cross-sectional studies, they tend to do the same, where looking for markers of aging, you grab people of different ages, and you say, what markers are changing over time, usually with linear regression or models like that. But if you think about it, especially as you get older, you realize periods of your life don't seem to be aging linearly. That is to say, you will hit, and we know this from a disease standpoint, as people hit their 60s, right? That's when diseases really escalate. And so what we did was we broke this down in a little more detail, where we actually looked for changes across periods of time, and both from a cross-sectional and a in the same person fashion. And it turns out most of your markers will shift, if you have enough data, will shift over time. And so, and then if you ask what molecules are changing when, most molecules, and you will age throughout your life, don't get me wrong, but there are in fact two periods that have bursts of changes, if you will. And intuitively, some of you will say, yeah, that makes sense. But one is in the mid-40s, and the other is people start hitting their 60s. And 60s makes sense. We know that's when disease risk goes up a lot. But it turns out there's a ton of biochemical changes that occur as people hit their mid-40s. And then you can, of course, look at the pathways that are changing. Yes?
You can say male and female, or is it different?
Great question. Yeah, it's not just due to menopause. I thought when we separated out the females and males, that'll separate that out. It's not the case. Males will be changing a lot in their mid-40s too, so you're not clear. All right, so the bottom line is you'll see that skin and muscle, and again, some of this intuitively makes sense, are shifting at both times. These are the pathways that change mid-40s. These are some of the things associated with that. Immune goes off in the 60s, carbs in the 60s. Cardiovascular disease, actually, believe it or not, changed at both. Not exactly the same molecules shifted. This will come out soon, so you can dig into the data in more detail. And kidney function, for example, in the later years. So you can see what things were changing when. And again, I think potentially this is actionable if you think about the kinds of pathways that are changing and oxidative stress and such. So one question I get asked a lot is, well, how are you going to scale this? And being in Silicon Valley, of course, the answer is you form a company. And so we formed this company, QBio, a number of years ago. They do a medical version of what I showed. They don't do RNA-seq and these things because it's harder to act on that information. But they also do whole body MRI. Now, if you ask any physician today, should you do whole body MRI, 100% of the time they'll say, absolutely not. And do you know why?
Expensive.
Expensive. What was the other? False positive. Yeah, I guess that's one way to look at it. They will find nodules, is what I like to say. Whether they're false or not, they're false for the moment, yes. So anyway, the point is, yes, you'll find these nodules. And how many of you have had a whole body MRI? This is probably, decent number. So anyway, if you ask me, I would say 100% of the time you should do a whole body MRI, because the issue isn't, do you have nodules or false positives? The issue is, do you have any nodules that are growing? And the only way you know that is through longitudinal profiling, right? You won't get that from a single time point. And so this group, this is from the first 100 plus people, they basically found, someone with early ovarian cancer, prostate cancer, even early pancreatic cancer, which is almost never found early. Okay, that was picked up by the MRI. And usually there's multiple lines of evidence that revealed these things. You'll see blood markers going off as well as imaging. And so again, this is very useful information, we would argue. And so again, obviously I'm conflicted on this one, but I do think this will be the future where we do these kinds of measurements. So the other area we've been spending a lot of time on is this idea about remote monitoring, Amazonizing healthcare, so to speak. And there's two approaches we're using for this. One is wearables, and the other is home microsampling, where you take little drops of blood, mail them in, and we measure thousands of analytes. I know what that sounds like, but ours actually works. Okay, so anyway, for wearables, there literally are over 1,000 devices out there, mostly in the forms of watches, some rings. I myself normally have 4 watches, one broke. I do have my ring. I normally have hearing aids, which are also sensors too, but I lost those. I gotta stop losing those. Anyway, they're expensive as well. But anyway, they're powerful because they measure you 24/7. They measure some pretty important things, right? They'll measure heart rate variability. But the power from them comes because they're measuring you continuously. So they're following you passively, so it's very easy to see when things go off. And so a number of years ago, when these first came out as fitness trackers, we said, well, let's try these as health monitors. So we put them on our cohort. And right away, we discovered you can tell when people are getting an infection from a simple smartwatch. And in this case, we discovered this when I first got my Lyme disease. It was asymptomatic, but I basically, at least when it first appeared, I was basically on an airplane at the time when it first became apparent. Maybe the backstory, just to fill you in, was I was in rural Massachusetts helping put up fences where 55% of ticks are Lyme infested. I was flying to Norway and you can tell I measure myself a lot with my smartwatch at the time, and I had a pulse ox. And you probably know already your blood oxygen drops on airline flights, why you get tired. But in this flight, I dropped abnormally low for that kind of a jet. And I didn't come back to normal when I landed. I saw my heart rate was up. I later learned my skin temperature was up, all pre-symptomatically. I was flying to Norway. When I did land, later I got a fever off and on. I went to a physician, warned him it might be Lyme. He drew blood, said, yep, your monocytes are up. You've got a bacterial infection. Wanted to give me penicillin. I said, no, I need doxycycline. Got a little tense because most physicians don't like their patients telling them what to do, but he did give in. So I took it, cleared it right up, and when I got back, I tested. I was Lyme positive, and I'd given blood before I left, and I was negative, so I seroconverted during that time. Very well-controlled experiment, actually. So in the end, it showed that you could detect, this is all pre-symptomatically when I first got Lyme. So I had 2 years of data at the time, so we looked at the data, and I was ill 4 times, and every single time, one was the Lyme, 2 were viral infections, the 4th was an asymptomatic time, but it had high, it's called C-reactive protein, which is a sign of illness, and every single time, I could see my smartwatch resting heart rate went up in advance of symptoms. So we wrote an algorithm to detect that. It worked on me. It worked on 3 other people who got sick, one of whom got sick twice. And every single time, we could tell when people are getting ill from a viral infection or otherwise from a simple smartwatch. And so we published that in 2017. Of course, along came COVID. Hold that thought. Good question. So along came COVID, spring of 2020. We're in a phase right now. You may know there's a new variant running around. And if you think about how do we detect this now, we use temperature, which is a 300-year-old concept. It doesn't work very well for COVID, right? Half the people don't get a fever. So it doesn't even make a lot of sense. Antigens and PCR tests are pretty good, but obviously you don't do them all the time. So we wondered whether we could use smartwatches for detecting COVID. So right away, when it first erupted, we partnered with Fitbit. We had 32 people who were wearing their Fitbit while they got COVID, and they had a symptom day and a diagnosis day. And so this is our very first case. This is resting heart rate. In purple is their diagnosis day, and red is their day they first had symptoms. And you'll see their resting heart rate jumped up 9.5 days prior to symptom onset. Okay. And in the end, from the 32 people, we could detect the onset of symptoms, the onset of this jump up in heart rate 80% of the time prior to symptom onset. And the median was 4 days prior to symptoms. So it turns out COVID has long pre-symptomatic periods, so it's almost ideal for this. And then we wrote a real-time detection system where you follow people in a circadian fashion, look for a jump up a resting heart rate for too high, for too long, and it sets off red alerts. And this does work. Again, here's one of our first cases here. This person, you guys can all sign up for the study. Whoops, it's on the previous slide. I'll send around an email if you like. But basically, this person first got symptoms here. They're diagnosed positive the next day, but they're getting red alerts for 3 days prior to symptom onset. And we know it works for Apple. We know it works for Fitbit. It should work for all devices that are out there. It's mostly an anomaly detection system. It looks for this jump up. And it works even in asymptomatic cases. Here's a Fitbit case where someone was diagnosed here, but they were getting red alerts for 2 weeks prior to symptom onset. And again, it works for Apple, it works for Fitbit. And it's even more sensitive than an antigen test. This is actually me. I had a particular family stress. You'll see there are other stressors that can give red alerts. And basically, the bottom line was I was getting ready for a trip to go to New York City. I woke up, I'm a little congested. You know that twilight zone where you're not sure, am I coming down with something? It was April, is it allergies? That's a real date there. And so I wasn't sure, so I got my COVID test. I was negative. I looked at my smartwatch and I was positive. What I do, I got on the plane anyway, went to this meeting in New York City. To go to the meeting, you had to get tested, at least back then, and sure enough, I was bright positive next morning. So I spent a whole week in a tiny little hotel room in New York City, all because I listened to my antigen test and not my smartwatch. Okay, so that also brings up another issue, which is it's not specific for COVID. So if you look at COVID negative cases, the number one trigger of red alerts is workplace stress. We have people annotating these things now. That's why we're trying to improve this. So we think it's also a mental health detection. Now I know this is why we want to pull in more data types. It was done early, where I'm pretty confident we should be able to tell the difference between a mental health stressing event and a respiratory viral one, because your respiration rate should be different if we could pull in those kinds of data. That's why I'd love to have you all sign up. Anyway, other things will trigger it too. Other infections, of course, will trigger it. And whether we'll ever be able to tell the difference between influenza and COVID, I'm not sure. We'll just have to see. But nonetheless, it is a sign that something is off. You could argue these are false positives. They're real signals, but what's causing them is not always clear. Although I would argue if you're just sitting around not doing much and you get a red alert, you're either mentally or physically stressed, one or the other. I'm pretty confident of that. All right, so we think this is the tip of the iceberg of what you can do with the smartwatch. If you think about smartwatches, right, they shine light into your blood. They're looking for spectroscopic shifts when they're measuring blood oxygen. And so with the 2016 watch and machine learning, we could actually pick up these clinical signals like red blood cell count, hematocrit, et cetera. These signals are not as good as clinical grade signals, but they're plenty good enough to detect a shift from baseline. You can see when things are shifting off. Even fasting glucose and hemoglobin A1C, you can pick up with a smartwatch. And so we think, and there's subsequent studies that also suggest this even now better. So we think these devices could be very powerful for measuring a variety of different conditions. The other area that we've been pushing pretty hard on, and I'll show you some new data here, is basically glucose monitoring with continuous monitors. So these continuous glucose monitors, they measure your glucose every 5 minutes. How many of you have worn one of these? Yeah, a lot in this crowd. They're really powerful, right? You'll never eat the same if you wear one of these. And I think in a good way, by the way. So anyway, I guess our claim to fame was we started putting these on so-called normal people and pre-diabetics. And what we discovered is that some normal people do have good glucose control, don't spike much. Some are moderate spikers, and some are severe spikers, just as bad as diabetics. Okay, so we wrote algorithms and classified people into glucotypes based on their spiking and glucose levels. And then what we discovered as at Aaron Siegel is that different people spike to different foods. So some people spike to pasta, others to potatoes, some to white bread, some to brown bread. It's all over the map. And Aaron's lab has gone on to show that about 21% of this is due to your microbiome. So here's some work from his, or some data from his work, which is here's an individual that spikes a banana but not a cookie. Probably the only person who doesn't spike to a cookie. Everybody spikes their cookies. I'm glad they didn't have muffins out there. I hate these diabetes meetings where they have muffins.
Is that controlled for total carbs?
Hold that thought. Great question. So here's a more typical cookie, but not a the banana. That's much more common. And we had found that some people spike to bread and peanut butter, others with protein bars. A lot of protein bars have carbs in them. Don't let them fool you. And then 80% of people spike to cornflakes and milk. That stuff is super evil, very nasty, probably worse than smoking. If you don't remember anything from this talk, I'd say don't eat cornflakes and milk. So to your point about carbs, so here we fed 50 people the exact same amount of carbs first thing in the morning while fasted, and they looked at their spiking patterns. And so what we'll see is that this person here, their top spiking food is bread. pretty reproducible, about 0.8 correlation between when you reproduce. This person's a grape spiker. This person's a pasta spiker. That person's a potato spiker. That person's a rice spiker. Most people are rice spikers, by the way. Rice is very, very common as a spiking food. So same amount of carbs, but different spiking to different types of food. So if you think about diabetes, it's really a complex condition, probably with underlying mediating causes. So your glucose control, it's mediated by your liver, your pancreas, your muscle, even your brain consumes a lot of glucose. All these things are all playing into your glucose regulation. And so there's different heterogeneity that you can think of at different levels when you think about diabetes. Some at the mechanistic level, like muscle insulin resistance, beta incretin effects, beta cell defects. Others would be, you can think about the biochemical pathway, what's off, and so on, and even the treatment pathway. Some people respond to some drugs, others to other drugs. So we actually dug into this a little bit deeper. I won't go through the assays, but we run these various assays. Well, maybe some of you know them, OGTT, SSPG, for actually determining things like muscle insulin resistance, beta cell function. Turns out I'm actually a beta cell defect, incretin effect, hepatic insulin resistance. So we can measure these with some of these gold standard assays. This is done with Tracy McLaughlin. And here's hemoglobin A1C, which is normally how we classify people's glucose dysregulation. And so at the end, you'll have people with different phenotypes, like each row is a different person. Each column's a phenotype. So this person over here, they actually have an incretin defect. That's their top defect. Most common is actually muscle insulin resistance. You'll see quite a bit of that here. Hepatic resistance and beta cell defects do appear as well. So we can quantify this with these gold standard assays. And then you can ask, going back to the point I just said, well, do any of these things correlate with the foods people eat? Well, it turns out if you're insulin resistant, you're a potato spiker. Okay, on average. There are individual differences, so don't assume anything is, you're the general person. Whereas insulin sensitive people, they don't spike so much, but you'll see even insulin sensitive people will spike the rice. So rice is tough on people, white rice especially. So anyway, so we're starting to tease apart the type of defect you have with what it means for your food consumption. And then we've been taking this even further. We're actually collecting a lot of information around people's lifestyle after having measured their underlying, if you will, metabolic subphenotype, their defect. So we'll collect information about sleep, physical activity, diet, not just the information about what they eat, but when they do these things. And what you find is that, so we get time-stamped information, is that you can start building predictive models around people's behavior and actually the phenotype they're associated with. So a simple one is over here, muscle insulin resistance is inversely correlated with the amount they exercise. So the more exercise, the less resistance you have, muscle resistance. But you can make other things as well, including the amount of carbs and other parameters affect like your adipose insulin resistance, your incretin effects, and here's a predictive model for even hemoglobin A1C. And then we can bring this, we think, into personalized management systems where if you're tracking people's lifestyle, and I'll come back to this at the very end, this is more at the general level. This is very complicated, but you can look at, for example, when's the best time to exercise? Is it morning or afternoon? Any guesses? How many think it's afternoon? How many think it's morning? Midday. Yeah, the data out there will say it's midday or afternoon if you look at the general population. But if you look at our study, it'd say, well, it depends on whether you're insulin resistant or sensitive. If you're insulin sensitive, you actually have better glucose control if most of your activity is in the afternoon. If you're insulin resistant, you have better glucose the next day if your activity is in the morning. So it's very personalized. Okay, and so we think this is how you can build personalized models around people. And then I'll just say, here I'm 100% conflicted, but I have a company called January AI that hasn't done exactly what I just said, but they are using an app. They have a 32 million food database, and they make personalized recommendations about what you should and shouldn't eat. If you wear a CGM for 4 days, they can make very useful predictive models. If you are going to go eat that muffin, it'll tell you to walk briskly for 15 or 20 minutes to suppress your glucose spikes. You can get very precise about it. And with their app, they've actually been able to show people improve their time in range for glucose and even lose weight. The last thing I want to tell you about, I think we have time for this, is this idea about home microsampling. So, why do you go to a physician to get measured all the time when you're healthy? You may come back sick after you get affected by someone else who's not healthy in the physician's office. So we spent a lot of time on this, actually doing what we call home microsampling. And there are two versions of this. One's off your finger prick, and the other's off your shoulder. We collect little drops of blood. You mail it overnight to the lab. And in the initial form that we published, we measure about 2,200 analytes. Now we can measure about 7,000, so much, much steeper. And we'll do proteomics, metabolomics, lipidomics, and very targeted assays. And I won't show you all the controls here, but most proteins, most metabolites are pretty stable. Some aren't. We know which ones aren't. Lipids, it's a mix. Some are, some aren't. And again, we know which ones are, which ones aren't. And so we can actually then run studies with this. In fact, most of our studies are now being run remotely, which is more convenient for people. It's cheaper, and they're in their more native environment if you run these studies from home rather than have them come into a clinic to get blood. So anyway, we did a couple of fun studies. I'm going to tell you about two of them. We had 32 people drink this shake that you can buy in CVS and lots of places called Ensure. And we sampled them. They're fasted. So folks are fast, and we sampled them before, 30, 60, 120, 240 minutes after drinking the shake. And then not surprisingly, hundreds of molecules change after drinking the shake. Oh, sorry, didn't mean to make it hard on you. All right, anyway, then we measured, as I say, all these, at the time, 2,200 analytes, and we can measure some pretty important things. That's your insulin C-peptide, your insulin. This is this incretin that's catching a lot of attention from Ozempic analogs and such. These are all hormones that affect glucose control. We can even follow inflammatory hormones. And the bottom line is, we can measure precisely in individuals. And so what we discovered is that everybody reacts to the shake differently. Same shake, very different response. So what I'm showing here are 3 different carbohydrates. Each box is a different person. And this is relative molecules, if you will, ion counts. So for this person, their carbs plummet after drinking the shake. For this person, they go up, 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 up. The person's flat, that one's down, down. Okay, same shake, different response. So we can cluster people informatically into 5 groups. And then these are classes of molecules. Gray is the average. The interesting one is brown. Brown is the inflammatory markers for folks. And so for this group of people, their inflammatory markers goes down when they drink the shake. For this group of people, it goes up. Same with this group of people, up. Same shake, pro-inflammatory on some, anti-inflammatory on the others. 10% of people have inflammatory bowel syndrome, and most of them have no idea what's causing it, but we could actually measure potentially in these individuals. Then the ultimate experiment is this one here, where we had one individual give a sample every waking hour for 7 straight days. Okay, and they're wearing a CGM, a smartwatch, food logging, and activity tracking. And not surprisingly, thousands of molecules change. And then the goal is to see which molecules change with activity and food to see if you can see what's going on and develop personalized responses. And we do these time lag correlations so that we can try and see which events precede others. Those are more likely to be causative events. And this is not a surprise, but we find thousands of associations between heart rate, glucose, and activity steps. In magenta here are the cytokines. These are lipids, and green are actually metabolites. And I'll just show you 2 examples here, but this is your insulin C-peptide, goes up 10 minutes after glucose. That's not a surprise. Here's kind of a cool one, alpha-synuclein, which is evolved in dementia and Parkinson's. It also shows an interesting pattern. We think it's going up in response to stress. So imagine you're at risk for dementia or Parkinson's. You might want to know this. The idea is, there's a hypothesis that we would actually try to control the spiking of this through behavioral intervention, such that maybe you could delay dementia for a few years, perhaps much like you might delay diabetes by knowing your glucose control and managing it. That's the idea. So there are many other interesting markers that were found in the study as well. So what we've done now is we built a personalized management system called MyPhD, pulls in all your wearable, your omics, your microsampling data, and you can display it whatever time resolution you want, daily, weekly, monthly, what have you. And then ultimately, this is the goal, it's to build personalized management systems. So this is the world I'd like to see, where people get their genome sequenced before birth, and then together with biochemical and wearable data, you build personalized AI management systems around people. And they really can just be individualized for the most part, because we all are pretty different. So these are the companies we've spun off from this kind of work. As I say, I'm a believer academics are good at proof of principle discovery. We're no good at scaling. And so this is what we've done. We've basically spun off companies, and the ones in boxes spin off from this work. And then just to show you one of these, this is the microsampling company called Iolo. What they do is you collect drops of blood from your shoulder, you mail it in. They measure about 600 metabolites that cover roughly 20 wellness categories. Things like oxidative stress, I guess I should look here, oxidative stress, inflammation, heart health. You can see the various categories. I've shown 6 there. And relevant to this group, you can actually track how people are aging relative to others in the cohort. We have quite a few people have now taken this. You can see how your biological age compares with your chronological age. But the nice thing is you can break it down into these age types. So for example, this person, their inflammation's pretty good, their metabolic age isn't so bad, but their heart age is up. And so again, it puts things in the categories, and you can use AI to draw in information and give recommendations that are not just exercise more, eat better, but based on the information you feed it, it says, eat this, don't eat that. It can be much more precise, and I think that's where everything's going to go. Everything will be managed by AI in the future. And so, in summary, what I've told you is that we think deep data does enable early disease detection, get a much clearer picture of people's health. We can follow people's age types and get at the genetic basis of disease using AI. We do think smartwatches are very powerful health monitors. Glucose monitoring, we think, is going to be very, very powerful. I forgot to say, but 9% of people in the US are diabetic, 33% are pre-diabetic, and those numbers are getting worse. Nearly everybody's pre-diabetic will become diabetic, so we think knowing this is important. And lastly, we told you about microsampling. And so I have an amazing team here. This is the Wearables team when it's at its peak. This is the microsampling, but I think I'll wrap up so I can ask questions if you have any. Answer questions, I mean. Sorry. Andrea.
That was amazing, thank you. So I have a couple of questions. The first one was, do you guys have any data or any association between the microbiome of the people that are different responders to glucose? Because it was a huge difference. So one can speculate maybe something is happening during the absorption phase that is kind of affecting how they respond.
Yes, we're collecting those data now. I don't have a story to tell you just yet, but maybe the next time you see me. Yeah. So that should account for some of the variation, but it's not going to account for all of it. I do think there's genetic, and what's very ignored now a lot is the epigenetics of all this. So I think that'll be a big factor too.
So quick question. I like to, we all here probably like to track this. This is your target audience. But I get frustrated because if you wanted to track all these things, you probably have to sign up for 15 different services. They don't talk to each other, and they don't talk to your physician who actually has to approve you to order any kind of intervention. And that's extremely frustrating. Do you have any thoughts on how we can pull this together and make it actually easy for people? Because it's almost a full-time job if you actually want to do all of this stuff right now.
Yeah, you're talking to a guy who's made it a full-time job. So totally agree. I think these things will converge to some extent. Like QBio, they did a medical version of this that you can take to your physician and stuff, and they're now bringing in the wearable data and things like that. So I think things will converge. Part of it is we don't know for sure what's actionable, and there's the whole price. This is another big factor, which is right now you have to pay out of pocket for all those stuff because that's part of our broken healthcare system. Nobody pays to keep you healthy. So we we really do need to change that. We need to change the financial incentives so that we can get this all aligned. But right now, people have to pay, and so there's this balance between what's the most bang for your buck. The deluxe form probably is a QBio. That's $3,500. They give a lot of medical testing along with a whole body MRI. And I hope to get that a lot cheaper, and I hope to add more data onto that as we know how to make it more actionable. And then some of these things, they are much more researchy. The smartwatches, the commercial devices, are not FDA approved. So by their very nature, they're going to have to come in through a different angle than a clinical one. So, but I do think ultimately this stuff should converge. I guess I view it as we're at very, very early days. And so I'm optimistic that we'll be able to do a QBio for, $300 or something in the future, and that somebody will pay for it because it saves them money down the road. That's my Pollyannish view of all this, but I don't know if that'll be reality. Yeah.
Yeah.
I think, sorry, we'll get you next if that's all right. Yeah, Vadim, sorry, I didn't see you over there.
So several of the parameters that you measure, like blood pressure, temperature, and other things, they change during the day, and you use them to estimate biological age. The question is whether biological age changes during the day and when we are youngest and oldest during the day.
It's a great question. We're poring over our digital data to try and get at some of that right now, like which variables change with age of those groups. And there's a paper up on bioRxiv that's looked at some of this from the UK Biobank. But I think there's a lot of information around, we tend to do things first thing in the morning. And I think there's a ton of information from the circadian values, because that does, as you point out, change with age. But I also think there's a ton of information around challenges. Like, is the best time to measure your health really first thing in the morning, or is it after you do a brisk walk? Like a stress echocardiogram, as you know, is after you run on a treadmill, and it's a great way to tease out a heart defect. And I like to say airline flights are free pulmonary tests. So we actually could collect a lot more information if we were to collect it at the right time. So you're raising a really great point that I think we should all be doing. And it's just a data collection and then an analyst problem. So I think it's solvable.
So to follow up on Mark's question, you give these talks a lot, and you've been doing this for a while. The current state of sites like QBio, of companies like QBio, is that they are not replacements for primary care physicians. And primary care physicians, for the most part, don't know how to deal with them. So you mentioned they've got this package that you can show to it, but so two sides of the same question. One is, how should people who are on the vanguard, like this crowd, who have a lot of devices, find primary care physicians who are in the know and willing to look at some of the kinds of data you're putting out there? And on the other side, do you, as part of your regular speaking, find physician communities to talk to instead of these sort of biotech communities?
Yeah. That's a great point, and the answer is they're almost always concierge physicians who charge 20 or 40 or sometimes even $250,000 a year.
Many of those don't know.
Yeah, they don't. They reach out to people like me or get advisors of people like me who will advise them on that stuff. But that's really where you get a lot of this. I think it comes down to, again, probably Pollyannish, that we're early days, and that if we can pull in this stuff. Like QBio, they didn't bring in wearables right away, and I like to think they're pretty enlightened. But they said, look, it's hard to bring that data in and get it back, because physicians will review their data, and they want to be, it's a very regulated space. As you know, 23andMe is a good example. If you start going outside the boundaries, you will get shut down. So, and there are some reasons for doing it. Is it over-regulated? Probably most people in this room would say yes. But I think, given that we do live in this regulated space, the information has to be vetted in a certain way and come in a certain way. And right now, people straddle it. Like YOLO, they call themselves a wellness company to get around some of this and pull in, I should probably watch what I say, but to pull in data, and people then can pull it in ways that they can use it a little more.
You're the one blurring the lines. And
We're trying to.
wellness
Yeah.
stuff, diagnostic stuff.
Yeah, I think in the end, if we can demonstrate utility for these things at whatever level, I hope the regulation will sort itself out. That's the hope. So first, show utility, and then get people, like genome sequencing when it first came out, I don't know how many of you remember these days, physicians were very adamant this was a bad thing to do, even for disease cases. I had someone who was in the neighborhood who's an oncologist. Oh, you must be sequencing genome. He said, no, nobody knows what to do with that. It's not that accurate, blah, 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 because he didn't know what to do with it, right? He wasn't really educated. And now, of course, if you have lung cancer, you don't get some genetic information. I would argue that's malpractice because there's 5 different treatments. They all depend on what your genetic basis is. So we just have to demonstrate utility, get it incorporated, and it takes time. We'll try and accelerate it if we can demonstrate utility. And actually what helped on the genome sequencing was people clamoring for it. So this crowd saying, I want that. And then that helps draw it in, and I think helps push the boundaries. So keep pushing, folks. I'm not sure. Oh, microphone. I got to follow the microphone. Yeah.
Yeah, so we have been doing so many different testing, but there is the utility the part that we need to prove of that. So what are you doing right now in terms of after doing the test? What is the next step of doing personalized intervention? Is there a recommendation system based on it? Like, what are the things that you are doing right now or you think is the next step?
List of, I need to do a Brian Johnson and put my stuff up there like he does. Of course, I'm always testing myself, so it shifts quite a bit. But I have my 10 supplements. Because I'm diabetic, I am on a GLP-1, and they work amazing. Now, I don't want to lose weight. You can probably tell losing weight's not one of my goals, in fact, but it does cause muscle mass, which I don't like. Yeah, so I am on some medicines. I'm on something called Farxiga. That's an SGLT2 inhibitor, again, for my diabetes. But they also have heart benefits that go beyond just the glucose control. So those two are probably and I take baby aspirin. Then I take a ton of different supplements, the usual antioxidants and anti-inflammatories and fish oil. And the same thing's probably half this room, probably 3/4 this room. So I guess I do those things. I do, and in January, I'll tell you the same thing. It's very clear you can keep your glucose level if you take a walk after dinner. Don't eat for 3 hours before you go to bed. Things you've heard about, but I'll tell you, when you see the data, they really punch you in the face, right? They tell you, don't do that. You wear a CGM and you eat cornflakes and milk, I don't think you'll eat cornflakes and milk again. It's just it's very visual. So what else do I do? I'm not sure. I had to make a list, but I gave you some of my obvious ones. Do take a walk. For me, I get very, very sleepy now after lunch because I have such strong glucose dysregulation. So I try to always take a walk if I can, especially if I'm going to eat anything.
So this is kind of an idea that I want feedback on from you, actually. So Carl mentioned the frustration we have with finding a physician that's actually, useful or understands any of this. I think everybody has that frustration. What about the idea of holding an annual competition where you try to get software to diagnose somebody, top physicians to diagnose somebody, and then a random sample of physicians to try to diagnose somebody and see who does best? And when software beats the physicians consistently, like that might shame the sort of healthcare system into maybe making some adjustments. I think this could be a really cool project, even if they don't react, at least.
I think it's a great idea. Do the physicians get to use AI or not?
Well, potentially, yeah, but they should. I would want them to be in their normal behavior that they would be doing in their practice, which is usually they spend 5 minutes thinking about a given patient.
No, and I'm empathetic with them. You've all been to a doctor's office, an appointment scheduled for 30 minutes, a nurse sees you for the first
It's more to
5.
put pressure on the system, not them specifically.
Yeah, no, it's an unfortunate. And, as in China, and the time a physician spends with you is 4 minutes if you're lucky. So it's even worse. So, it's a problem. And this is where AI should help a lot because it should be able to pull and integrate information so much better than a physician can do. So in the future, we all want a physician who uses AI versus one who doesn't. And this idea about waiting for perfect AI is crazy because most physicians are a long ways from perfect. So I think, yeah, we need to bring as much information as most useful way as possible. I love your idea of a competition. Yeah, be pretty cool.
One for driving.
Yeah. Anyway, thanks again.
Thank you.